Meiosis is very similar to mitosis. If you don't know what mitosis is a form of reproduction for cells that only has one cell division and is used to make more somatic, or body cells. Meiosis, on the other hand, has 4 cell divisions and is used to make gametes. Before meiosis starts, the cell is an interphase. In interphase, the cell is growing and doing all of its normal activities. But we're more interested in interphase S. In interphase S, each chromatid is replicated so that each chromosome has 2 chromatids instead of 1. Now that all the chromatids have been replicated, meiosis can finally start. Meiosis is broken up into 2 phases, meiosis 1 and meiosis 2. Let's talk about meiosis 1 first. The first phase of meiosis 1 is prophase 1. In prophase 1, the chromatin begins to condense into visible chromosomes. The nuclear membrane will also begin to dissolve, and centrioles might have started to form by now. Each chromosome tries to find its homologous pair. Homologous pairs are 2 chromosomes that are about the same size and shape. More importantly, they contain the same genes. When 2 homologs meet, they get really close together and form an overlapping region called the chiasmata. While they're here, they'll start a process called crossing over. Crossing over is when 2 homologs exchange pieces of their genetic code. This is completely normal and will result in more genetic diversity in the 4 daughter cells. It's important to note that the cell right now is diploid, which means it has both its paternal and maternal chromosomes. Now metaphase 1 begins. In metaphase 1, the chromosomes move to the equator, or center, of the cell in 2 lines. The centrioles migrate to opposite poles and spindle fibers form between the centrioles and the chromosomes. After the chromosomes are pulled to the middle of the cell, anaphase 1 begins. In anaphase 1, the newly formed spindle fibers shorten to pull the chromosomes away from each other to the centrioles. Then telophase 1 can begin. Telophase 1 is the first cell division in meiosis. The centrioles and spindle fibers dissolve away, then the cell starts to stretch before finally splitting in half. A new nuclear membrane is formed around each group of chromosomes. As you might have realized, each of the cells after telophase 1 has half as many chromosomes as the original cell. However, each cell still has the same number of chromatids, so by the end of meiosis 2, another cell division will occur to decrease the number of chromatids in each cell. Compared to prophase 1, prophase 2 is pretty boring. All that happens is the nuclear membrane breaks down and the centrioles reform. No pairing up with homologs or crossing over occurs in this phase. In metaphase 2, the chromosomes, once again, line up in the middle of their cells. However, they only line up in 1 line instead of 2. The spindle fibers also reform between the centrioles and the chromosomes. Anaphase 2 is a little bit different than anaphase 1. Instead of the chromosomes just being pulled to one side or the other, they actually break apart at the centromere. Centromeres are just the center of a chromosome where 2 chromatids are attached together. Other than that, it's exactly the same, with each chromatid being pulled to the centrioles of the cell. In telophase 2, the centrioles and spindle fibers dissolve away. Each of the cells divides again, forming 4 daughter cells. Each of these daughter cells is haploid, which means they have half as many chromosomes as the original cell. Then the nucleus reforms. Meiosis is used to create unique cells, since each of the 4 daughter cells will have different chromosomes and genes than both the original and sister cells.